Hello everybody, this is Dr. Christopher White and welcome back to part 4 of the Precambrian and its life history, focusing on the Hadean and the Archean. Alright, let's get going. So in the previous lectures we've been talking about the geology of the Precambrian. So now we need to start thinking about life. So the first question is well, when did life actually appear? When can we begin to see the first indications of it in the rock record? Well, we have definite monera fossils. So monera is a single-celled organism, and that dates back to 3 billion years. Now that's a proper fossil. We actually have the imprint of the organism itself in the rock. And we have probable fossils back to 3.5 billion years. We also have geochemical indicators of life in the rock record all the way back to 3.8 billion years. So it's primarily focused on a layer of graphite-rich rocks which are part of the Isherwood greenstone belt. So we can say with certainty we definitely had life 3 billion years ago. We can say there is an extremely good chance we had life 3.5 billion years ago, and we can say there is the possibility of life 3.8 billion years ago. So we've covered evolution already, so we have some idea about what will happen once we actually have life. But the question is, obviously we need to, somehow make living organisms from essentially natural material. So how are we going to make that happen? So the process of abiogenesis is the method through which we take we make living organisms from non-living matter. So interestingly, abiogenesis is not a random process, as natural selection will actually function on some level, because some abiogenic processes will be helpful and some abiogenic processes will not be helpful. And so obviously the helpful ones will essentially move the process forward. Abiogenesis does not mean that we instantly have fully formed cells or complex molecules like DNA or RNA. They take a long time to get to. Instead, the process of abiogenesis is going to require billions of steps, and each one of these steps will increase the organization and complexity in very, very small increments. There will also be innumerable failures. So this is going to be a process that's going to be very, very complicated. However, this process isn't happening, in just one test tube or in a pond somewhere. This is happening across the entire surface of the Earth. So these billions of reactions which are taking place, when you think about it on a global scale, well, all of a sudden that doesn't become too difficult. And so, we're going to have billions of reactions happening every second. And so eventually, just through the process of, chance, we're going to slowly but surely move the process forward. So we need to think about when really is something considered to be alive. And there's 7 criteria we like things to have in order to actually be considered living. So the first thing is that your organism should be able to maintain balanced conditions inside itself. So that's called homeostasis. So for instance, a classic example of that is human beings have a set body temperature. As soon as we start moving either above or below that set body temperature, our body starts to get distressed because we are failing to maintain homeostasis. So an organism should also have a highly organized structure. This doesn't mean your organism has to be extremely complex, but it does mean it has to have some kind of organization to it. Your organism should be able to break down or build up nutrients, so that's a metabolism. Your organism should be able to grow, so its structure will change over time in an advantageous manner. Your organism should be able to show adaptation to its host environment. It should respond to environmental stimuli, and it should be able to reproduce. So what we need to try and do is we need to try and take essentially non-living matter and turn it into something that meets these 7 criteria. So if life is to originate, we need to begin by synthesizing the organic molecules that we need to build it. So luckily the elements available for, at the time were water, carbon dioxide, nitrogen, ammonia, methane, hydrogen sulfide, sulfur dioxide, phosphorus. So we actually have everything we need to make the organic molecules from which life on Earth is built from. So molecules on molecules that are used in the biological process on Earth contain hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus. So we have all the basic building blocks available right from the start. So we've already managed to pass stage one. So the organic molecules formed from these simple building blocks are called monomers, and something like an amino acid is a good example of a monomer. And these monomers are going to be the building blocks of more complex molecules, the kinds of molecules we need in order to build cells and to have those cells function correctly. So our first question is how do we start to come up with these monomers? Or more importantly, how do we make the amino acids that we need to build life? So in 1950s, a scientist called Stanley Miller took a gas mixture which was approximate to the composition of the Earth's early atmosphere, and he subjected it to electric sparks which simulated lightning. So here's the basic setup. So we have water here which is being heated, so that's going to push gas essentially through the piece of equipment in a cycle. This area here is filled with gases which approximate the Earth's early atmosphere. These two electrodes are going to pass an electric current between them to simulate lightning. And down here we have what's called the trap. So as the water comes around here, it cools down. The liquid water will then drop down this pipe and it will cool down, and it will settle down in the trap, and it will be swept through to replace the water that's lost from the boiling vessel here. So what happened was Stanley Miller ran this experiment, and after a little while he realized the water down here in the trap was getting cloudy. And so naturally he assumed that he hadn't cleaned his equipment well enough, so he took everything to pieces. He went and cleaned it up, and he went and did the same process again, and exactly the same thing happened. The water at the bottom of the trap here started getting cloudy. And he ran it a few more times, and the same thing kept happening. And so what he did is he collected the water from down here in the trap, and he analysed it. And when he analysed it, he discovered that this water was stuffed with amino acids. And so what was happening is the electric spark was sufficient to provide enough energy for a chemical reaction to take place to produce amino acids, which were essentially becoming part of the water down here. So over time, the longer he ran the experiment for, the more amino acids he had gathering down here in the trap. So updated experiments using the same setup and a slightly different gas mixture have produced nearly all of the 20 basic amino acids required for living organisms. So essentially we can make most of the building blocks that we need simply through this process. And the great thing is that there's lightning happening on across the Earth all the time. And so if you imagine, every day you're going to have millions of little lightning strikes which will create amino acids which will fall out of the atmosphere and become incorporated into bodies of water. And so obviously during the Archean, what was going to happen is you were going to start building up these amino acids on the surface of the Earth. So we've managed to achieve stage one already relatively simply. So good work, us. Okay. So you can also synthesize amino acids through ultraviolet light strikes, or maybe some of them could have been added by comets. So there's a few possible ways in which we can get amino acids onto the surface of the Earth. The most likely one, though, is the formation of amino acids through lightning strikes. Okay, so we've got our amino acids. The next stage is taking those amino acids, so the monomers, and turning them into larger molecules. So this process of taking monomers and stringing them together to make larger molecules is called polymerization. And the molecules you produce are called polymers. So polymers are made up of lots of monomers which are strung together. And a good example of a polymer is something like DNA or RNA. So small molecules, approximately 200 amino acids long, are called thermal proteins, and we can very successfully synthesize those in the lab without any kind of special process required. So how do we make these small polymers? Well, if you take a liquid, so a water, that contains amino acids and you begin to heat it up so the water begins to evaporate away, eventually the water makes it to the point where it's getting pretty close to have fully evaporated, and at that moment what happens is the monomers spontaneously begin to polymerize. So they'll start to join up and link together to make polymers. So all we have to do is we have to take ourselves some amino acid bearing water and heat it up. Now luckily this is going to be quite easy to do in the Archean, because we've already discussed the surface of the Earth in the Archean was hotter, so there would have been lots of volcanic activity pumping heat into the rocks. The rocks themselves of the crust would have naturally had more heat passing through them, so they would have been warm. And we also know that the environment would have been kept warm due to a very strong greenhouse effect, because we had all this carbon dioxide in the atmosphere. So this would have meant the Earth's very early warm environment would have been a great place for this process to take place. It wouldn't have taken much effort to have taken a body of water containing amino acids, evaporate most of it away, and right towards the end of that evaporation process, all of a sudden the monomers will spontaneously polymerize. So the process is modelled to occur at approximately 70 Celsius. That's when most thermal proteins form. And so this is a temperature that's pretty easily achieved in the Archean. So we've taken our next step with relatively little fuss. So we've managed to make our monomers, so we've made our amino acids, and then we can take a water containing those amino acids, we can begin to heat it up, and eventually those amino acids will polymerize, essentially, and will start forming small forming small polymers about 200 amino acids long. So these polymers, these proteins, were not synthesized by organic processes though, so they're considered to be artificial. So they're not really called proteins, they're called proteinoids. Now, the problem is that proteinoids will very quickly dilute if you leave them in water. So what we really need is we need some kind of protected vessel in which we can have these polymerization reactions take place. So to put it another way, what we really need is some kind of self-contained system like a cell. Now, it just so happens that if you have liquids or water that contains proteinoids and you agitate it, so you just stir it up a little bit, the proteinoids will naturally clump together to form these things, and they're called microspheres. So the membrane you can see here is made of the proteinoids, and so what you're looking at is a crude cell. So we have a crude protective vessel. Inside here, the material in here is protected from the outer environment, so this means chemical reactions that take place in here will be protected, so the materials that form will not be being diluted. And so this means that when you have lots of microspheres, they're going to create a soup of material inside themselves. Now, microspheres are able to split and they are able to combine together, but that's not really reproduction. Okay, all that will happen is they can split clean in two to create two microspheres, which are each half the size of the original, or maybe you can have two microspheres combined to form one microsphere, which is double the size of the original. So the great thing about microspheres is they offer a protected environment in which these chemical reactions can occur, but they will only typically last for seconds to days. They're not strong structures. But the thing is inside here we have these chemical reactions taking place, and when the microsphere does pop, it's going to release all of these products into the water. And obviously the water is being agitated, and so any proteinoids that are made in here are going to go to create more microspheres. And so obviously you can see the more microspheres you have will create more proteinoids, which will allow you to then increase the amount of microspheres you have in the next generation even more. And so each step we are steadily increasing the amount of microspheres, and each one of those microspheres is going to create more and more material inside themselves. So you can begin to see now we already have the next step required to start allowing us to have these chemical reactions which are going to bring about life occurring in quite a large process. However, if we really want to make this work, what we need is a much stronger cell membrane. We need something a lot more robust. So what we need are fatty acids or phospholipids. So I apologise for all the text on this slide. So when did fatty acids appear? Now this is actually quite a difficult question to answer. However, experiments have shown that when you produce proteinoids, you will also form fatty acids as a byproduct. And these fatty acids will naturally clump together on clay minerals. So we're actually looking at down here. So the nice thing about fatty acids is they're a molecule that has a hydrophobic and a hydrophilic end. So the hydrophilic end of the molecule, marked out here by the white circle, likes water. So it wants to be in water. The hydrophobic end of the molecule, on the other hand, does not like water. And so what happens is if you take a fatty acid and you put a drop of it into water, the fatty acids will naturally orientate themselves with the hydrophilic end pointing out and the hydrophobic end pointing in. So this is the kind of process you see when you drop something like olive oil into water. You will notice that the olive oil will form a nice spherical blob, and it's exactly the same process. The fats in the olive oil are naturally orientating themselves to form a similar pattern to this. Now what we have here then is we now have ourselves a relatively strong vessel, don't we? We have a wall made of these fatty acids, and in the middle of it here we're going to have a void in which chemical reactions can take place. So the fatty acids will naturally arrange themselves to produce what's referred to as a micelle, which is this thing right here. Now the fatty acids provide a stable membrane, and it will essentially protect the fluid within. So great, we now have ourselves a robust cell, a robust vessel in which we can continue our chemical reactions. Now what we really want though is we really want the formation of phospholipids, because phospholipids are what most cell membranes are made from now. And so phospholipids are kind of the next step up. So once we have large enough RNA molecule, so RNA is like DNA, but it's not quite the same. We're going to discuss it in a couple of slides time. So once we have large enough RNA molecules, that will allow us to develop enzymes. So an enzyme is a protein that acts as a catalyst. It helps to increase the rate and success of chemical reactions. And so once we have RNA appearing in our molecule, producing the enzymes required, we can start to convert fatty acids into phospholipids. And once that process starts happening, then we have the capacity to create these nice, super strong cell membranes made from phospholipids. And then once that happens, then we have ourselves a really solid, robust vessel in which our chemical reactions can occur. So the important point is once we have enough phospholipids, we manage to form something which is called a liposome. And you can see here, so we actually have what's called a bilayer sheet. So we have two layers of these phospholipids. So we have the phosphorus end, which is the hydrophilic end, and we have the lipid end, which is the hydrophobic end. And you'll see they'll orientate themselves facing away. So we have the hydrophilic ends pointing outwards and the hydrophobic ends pointing inwards. And so we end up with this void in the middle here. And this is going to be our crude cell. So the great thing about bilayer sheets and also the layers that we build up in micelles is that they will let small molecules in, but they will not let large molecules out. So we can have things like amino acids pass through the membrane into the interior, but anything that's created using those amino acids is not able to leave the cell. And so this means the cell, and we have chemical reactions taking place in here that are constantly being fed by new material coming in, and it's creating essentially a soup of proteins and polymers and lipids and other things inside our cell. And this is exactly what we need, because what we need is a nice protected environment in which we can allow these chemical reactions to take place. Okay? And once again, some of these chemical reactions will be helpful and some of these chemical reactions will be detrimental. Obviously, if they're bad chemical reactions, it will end up destroying the liposome, and so that will be natural selection. If, however, something happens which is positive and it helps to move the process forward, essentially that will then take the process to the next stage. So we have lots of monomers, and these are being polymerized into protective environments like microspheres, mice cells, and liposomes. So the next question is well, how do we start reproduction? So microspheres will divide, but they'll only divide by essentially splitting themselves in two. So you'll take one microsphere, you'll split it in two, and you'll end up with two microspheres which are half the size of the original. So that's not true organic division. Now, micelles and liposomes, on the other hand, are slightly different. So we've already discussed how material is going into the micelles and the liposomes, but it's not able to get back out. And so this means that the volume inside your cell is going to have to steadily increase, isn't it, to make room for this new material. So this obviously means that we'll need to keep building out the cell wall. So your vessel will need to create new fatty acids or new phospholipids to allow it to increase the size of the membrane. Now, eventually you're going to get to the point where your micelle or your liposome grows so large that it's naturally going to become unstable and it's going to split in two, essentially. And the great thing is when this cell membrane fails, it will naturally seal itself straight away, because remember, the fatty acids and the phospholipids are going to want to close up as fast as possible. So as long as we can create the fatty acids and or phospholipids required to keep increasing the size of the cell membrane, we can make our vessel grow larger and larger and larger until it eventually splits, in which case we now have two vessels which are about the original size of the parent. So essentially we've managed to reproduce, haven't we? It's very crude, but we've managed to do it. So eventually, yes, a liposome will reach a point where the division will produce two micelles or liposomes which are going to be of a similar size to the original cell, the original vessel. Now, within the micelles or liposomes, polymerization is difficult without enzymes. So an enzyme is a catalyst. It's a long protein that helps to speed up the process and success rate of chemical reactions. So enzymes are very, very important to us. If we didn't have enzymes inside our bodies, we would be in a lot of trouble. However, enzymes are really, really big proteins, which are polymers. So we have a problem in that polymerization inside these micelles and liposomes is difficult. However, in order to actually make more of what we need for our cells, more fatty acids, more phospholipids, more proteins, we need the enzymes, which are massive polymers. So it's really difficult to make polymers, but what we really need are polymers. So we have ourselves a bit of a problem at this point, don't we? We could, we're heading very quickly, towards a brick wall. So we need to come up with a way around this problem. So how do we get around our enzyme problem? Well, luckily there is actually a relatively straightforward solution, and that solution is RNA. So what's RNA? Well, RNA, if you want to think about it in a very basic level, is a simplified version of DNA. So it's a molecule in which we will be storing information. And we store information in the form of base pairs. Now, in the case of DNA, we have 4 base pairs. We have G, that stands for guanine, C stands for cytosine, A stands for adenine, and T stands for thymine. And in the case of RNA, we have the same bases, with the exception of thymine, which is missing, and it's replaced by another base called uracil. So the way these bases are arranged essentially controls how the molecule works. So if we look at this molecule, this protein down here, what can we see? Well, each of these represents an amino acid, and the order in which these amino acids are being arranged is based on the RNA. So if we look, AUG, so those 3 bases together, means that this amino acid will be put in place. Then we have CUU, stands for this amino acid. UCG stands for this amino acid. UAU stands for this amino acid. So the way in which the bases are organized controls the sequence of amino acids and how they're stacked together to form longer proteins. And so the great thing about RNA is essentially it means you can create a protein, then that protein will essentially float off and do whatever it's got to do, and then the RNA will then create the same protein again and again and again and again and again. So it's a mechanism by which we can reproduce the same proteins repeatedly. And obviously if you need these proteins, so let's say you're putting together something like a cell wall, so you need fatty acids or phospholipids and or other longer molecules, the RNA allows you to reproduce these molecules in very, very large quantities quickly and efficiently. So essentially it's catalyzing these processes. So the studies we have indicate that RNA was probably what allowed us to get over this enzyme problem. It helped us to catalyze these early reactions which we needed to keep moving these protocells forward. So we need to remember, of course, that RNA and DNA are essential for the synthesis of more complex proteins. So the nice thing is once we have RNA, because the order in which these bases are organized would initially have been random, essentially it means we produce a whole range of different proteins. Now, obviously, some of these proteins will be good, in which case it's going to push the evolutionary process forward. And in some cases, the proteins produced could actually be harmful, in which case evolution is going to remove that particular protocell. So small sections of RNA have been made in the lab under what we consider to be natural conditions. So this suggests that the synthesis of RNA abiogenically is possible. So we can make RNA without the need for a biologic process. So we can just make it with the minimum of fuss. And this is good because once we have RNA, well, then it can start to help catalyze these reactions inside the cell. Now, the other great thing is that we also know that RNA molecules can reproduce themselves. So once we form a piece of RNA, it will copy itself several times again and again and again. And that means one piece of RNA can only do, let's say, one protein at a time. But because it can copy itself, you'll have hundreds of pieces of the same piece of RNA. That means you can reproduce the same protein numerous times in a very short period. So this helps to really push forward the rate at which we're making new molecules inside our cells. Now, one of the other good things is when the RNA copies itself, it's actually prone to what we call transcription errors, which occasionally means it makes mistakes. And so this is going to also help to add in an extra layer of variation, which helps to also push evolution forward, because as the DNA changes its base pair arrangement, that's going to change the type of proteins that form. And once again, sometimes one of those changes will bring about the formation of a new and helpful protein, and that's going to help to push evolution forward. So the final thing about these small pieces of RNA is we now know they can also join themselves together to form larger pieces. And so RNA is a beautiful fix to our enzyme problem. It helps us to get over it for a short period of time. It helps us get to the point where we can start forming larger proteins as we start to form larger pieces of DNA. And then once these larger proteins start to essentially form enzymes, well, then the enzymes can take over and help to catalyze the reactions even more efficiently. So studies have also shown that all the building blocks for RNA can be produced through the reaction of proteinoids with common minerals. So we can make everything we need relatively easily with the minimum of fuss. And we've also have more studies which have indicated that small pieces of RNA can actually join themselves together to make bigger pieces of RNA. And so by adding additional RNA pieces, what you do is you increase the functionality of your molecule. The bigger your piece of RNA is, the bigger the polymer it can produce. So as these RNA molecules become larger and more complex, they will fabricate larger and more complex proteins. And this will eventually lead to enzymes. And so once we have enzymes, we now have another tool in our toolkit that's going to allow us to essentially increase the rate of chemical reactions and increase the success of those chemical reactions. Now, once again, just like the earlier parts of the process, some enzymes which will be created will be helpful, in which case we've moved evolution forward. Some enzymes created will be harmful, in which case that particular vessel will be removed from the evolutionary process. So you can see natural selection is going to help keep pushing this process forward. The other great thing about RNA is that when it copies itself, it's very prone to what are called transcription errors. And this means that new RNA molecules may not be the same as the original. So an RNA molecule will spontaneously change if it has one of these transcription errors. And this means that what we're doing is we're providing heritable variation. So we're changing the genetic information, and we can, this changed genetic information can then be passed on to the next generation. So you can see we are now being able to pass these changes forward generation after generation. So this process means that during these early stages we'll form proteins. Sometimes some of those proteins will be helpful, some of those proteins will be harmful, and some of those proteins will be neutral. And as we've discussed, natural selection will kick in. It will remove the vessels that have the harmful proteins in them, and it will essentially push forward the vessels that have the helpful or neutral proteins in. So natural selection will allow cells with new and more advanced pieces of RNA to become dominant, assuming the RNA provides some kind of advantage. And so this is going to keep pushing the process forward step by step. So you're going to keep increasing the size of your cells. You're going to keep increasing the complexity of the RNA and the proteins you can produce. That's going to help produce enzymes which will catalyze the reactions inside the cells. That's going to increase the rate of reactions and increase the success rate. It's going to lead to the formation of new, more complex proteins, and so on and so on and so on. And so you'll see over time what we're doing is we're increasing the complexity of our cells. So by the end of the process, we've ended up with a cell that has a organized structure. So it has a cell membrane. It will change over time. It will adapt to its environment because it will, because it will change what chemical reactions it can do based on what's available. It has a metabolism, so it's taking material in and processing it to make new proteins, for instance. And it achieves homeostasis. The chemical reactions taking place will naturally equilibrate. So we've filled nearly every single criteria for something to be considered alive. The only outstanding criteria to be considered is the ability to respond to an environmental stimuli on demand, and that one is a particularly difficult one to quantify. So let's be honest, at this point we can see that we have a process that could work. Scientists agree on these basic steps, and so the formation of microspheres, micelles, liposomes as early cells, and the production of RNA and how that helps us to increase the complexity of the proteins produced, we can all agree pretty strongly that that would work under natural conditions. However, one of the big disagreements is the origin of the monomers and the polymers. So many argue they were sourced from atmospheric compounds, as we've discussed. However, some people actually argue that life started around hydrothermal vents. And so this is going to bring us on to submarine hydrothermal vents. So a submarine hydrothermal vent is produced when water, so seawater, gets pulled down into the oceanic crust. Because it gets pulled down into the crust, it gets heated up, it becomes more buoyant, and so it rises through the crust, and it will vent out onto the seafloor. And it will produce these things which you refer to as stacks or chimneys. So the vents are sometimes referred to as hydrothermal seafloor vents. So as the water passes through the oceanic crust, it will scavenge, it will dissolve elements from the rock. So it will steal things like iron, copper, lead, zinc and sulfur. And so what we have here is we have this chimney structure made of sulfide minerals. So it's going to be made mostly of the mineral pyrite, iron sulfide, but mixed in with it we're going to have copper sulfides, lead sulfides and zinc sulfides. So yeah, these elements are precipitated as sulfide minerals when the hot, high-pressure hydrothermal water enters the cold, low-pressure seawater. So this is our basic model. Here's our piece of oceanic crust here in brown. We have our oceanic water being pulled down along faults into our oceanic crust. It gets heated up by intrusions. The water obviously becomes hotter and more buoyant and wants to rise. as it's circulating through the oceanic crust, it's stealing a whole range of elements from the rock as it goes. And so when the water vents back out onto the seafloor, you have very hot water coming into contact with very, very cold water. And so this is going to encourage a lot of these dissolved metals to precipitate out as minerals. And so that's going to help to produce our sulfide-rich chimneys. So the mineral buildup forming chimneys and around vents can reach 40 to 60 meters in height. So some of these chimneys can actually be absolutely massive. Now, if the sulfide-rich hydrothermal fluids have a black colour, they are termed black smokers. So the black colour comes from typically large quantities of iron being dissolved in the water. However, sometimes submarine hydrothermal fluids will circulate through felsic to intermediate rocks. So remember, the oceanic crust is made of mafic rocks. Sometimes these hydrothermal fluids will circulate through rocks which are felsic to intermediate in composition. And so this means as they circulate through those rocks, they're going to scavenge, they're going to steal different elements. So instead they'll steal things like silicon, calcium, barium, and other dissolved metals like lead. And so that means when this fluid vents out onto the seafloor, it's going to have a different colour because the composition is different. So in that case, we're going to have what's called a white smoker because the fluids are obviously white in colour. You can see one of the chimneys right here. You can just see it there. So during the Archean, the Earth's interior would have been hotter and the lithosphere would have been thinner. So submarine hydrothermal vents would have been very, very common during the Archean. Now the thing about these hydrothermal fluids and the seawater, well should I say the seawater that is used to make the hydrothermal fluids, well it will contain calcium, hydrogen, oxygen, nitrogen, phosphorus in small quantities and sulfur in quite large quantities. And so once again we have our basic building blocks for molecules, so we can make our monomers. And of course we're in an environment that's very, very hot. So the water around our oceanic vent here is going to be a lot warmer than the surrounding seawater because our hydrothermal fluids are going to have a temperature of 350 Celsius. And that means all of this hot water being pumped into this environment here means the water around here is going to be quite warm. So you're going to have warm water and you're going to have the chemicals required for abiogenesis. So it's hypothesized that you can form the monomers required in the environment relatively easily. Once again, you've just got to combine the building blocks together, and you can do this because we have a nice warm environment. So, we've got lots of chemical energy around, or lots of energy around to help push the reactions forward. So once we have our monomers, you can polymerize them to make larger molecules. And it's been found that if you take amino acids, they will naturally polymerize on the surface of clay minerals. And it just so happens that in the deep ocean environment, there tends to be a bit of clay. And so this means that you will have the basic location that will allow you to bring these monomers together and polymerize them to form more complex polymers. So the polymers go to form protocells. So we are talking things like essentially microspheres. And analysis of hydrothermal waters has revealed the presence of amino acids. So we know in these areas there are amino acids around. We know they will naturally congregate on clay minerals and they will naturally polymerize. However, it's also been argued that this very hot, very aqueous environment, so very water-rich environment, may actually inhibit polymerization. Polymerization is not an easy process to occur naturally. Remember, we discussed the process we were using before was a thermal process. We were evaporating away the water. That doesn't work in this setting because although the setting is very, very warm, there's loads of seawater. So it's a very aqueous, rich environment. And so there's a good chance that will actually inhibit the capacity of the monomers to polymerize and form larger molecules. So it's uncertain whether hydrothermal vents actually offer a suitable location for life to begin. However, if they did, it would have a major impact for the idea of life in our solar system. So if we look at this, what we have here are some of the satellites, so the moons of some of the planets. So we have the Jupiter's moons, or some of Jupiter's moons here. We have Saturn's moons, so we've got Titan, and we have, there's two down here, we have the moons of Uranus, we have Neptune's moon, and we have some of the dwarf planets, including Pluto. And one of the things that's become clear over time is that these satellites have, and the dwarf planets should I say, have thick layers of ice. And these are going to be water ices, carbon dioxide ice, methane ice, so it's not just all water ice. But we also know that the interior of these satellites and dwarf planets is going to be warm. And so this warm interior is naturally going to melt some of the ice down at the bottom. And so we think there's a very good chance that under a lot of these outer icy shells, there is actually a body of water. And so all of a sudden we have ourselves a situation where we could foresee some of this water being pulled down into the rocks, circulated around, scavenging metals as it goes, and then being vented back into this ocean underneath the ice. And so if these hydrothermal vents do provide a good location for life to begin, well then all of a sudden we have multiple possible targets in our solar system where life could also currently exist. So we need to bear this in mind as well. So if we could work out that hydrothermal vents actually offered the conditions that would produce life through abiogenesis, well then all of a sudden there's the possibility that we may not be alone in our solar system. Now, in all probability, it's likely that life around submarine hydrothermal vents migrated in and adapted to the environment. That's the more probable situation. However, what can't be denied is that these vents offer an island of biodiversity in an otherwise pretty barren environment. So you can see the sheer quantity of life associated with these hydrothermal vents in an area which is very, very devoid of organisms. The abyssal plain in general is, for want of a better way of describing it, pretty barren. Okay, so this is a good place to stop part 4. So obviously stop the video, get up, have a walk around, go and get a glass of water, sit down, relax for a few minutes, and then please come back for part 5.